This is a short podcast on enzymes, and those of you that have the enzyme lab, this might be helpful in answering some of the questions about enzymes and how they work. So if we look at the beginning, enzymes are called catalysts. And catalysts just speed things up, speed up a chemical reaction, which we talked about in class. Enzymes are in your spit, and they help kind of get the digestion process moving. And so enzymes are just proteins that act as a catalyst in all living things, and it functions as a catalyst by lowering the energy needed to run the reaction. So any reaction that we have in our body takes energy to do it, and so enzymes just simply lower that activation energy. And because you're lowering that activation energy, the reaction happens a little faster. So there's a couple of parts. There's called a substrate, which each enzyme fits a specific substance called a substrate. Like if it's food that you have or a molecule you want to break down, that's called the substrate. And we call this the lock and key model. So for every lock, there is one key, and enzymes work the same way. There is one enzyme for every substrate. So the place where the substrate binds is, or the enzyme binds to the substrate, that's called the active site. We'll take a look at this in just a minute. So there's two ways that enzymes work. You're either going to break something down or you're going to put something together. So if you, in this instance, if you break it down, you have your enzyme, you have the active site here, you have your substrate, let's say food, and you want to break apart your food. Food binds to the specific enzyme in your spit, chemical reaction starts to take place, and boom, you got it broken apart into whatever product you have. Right? This would be like breaking down glucose in food. Food binds to enzymes. You run through cell respiration and you break apart the glucose to get ATP at the end. If you want to make something, we have another enzyme here. The active site is right at this spot. It's like a puzzle. And so you have two substrates that you want to put together. Let's say in photosynthesis, you want to combine carbon dioxide from the air, water from the ground, and using sunlight, we're able to have water, have carbon dioxide, chemical reaction takes place with sunlight, and voila, we get some glucose in the end, we get our product. So that's really how enzymes work. Now, most enzymes are reversible, okay, which all that means is that enzymes don't ever disappear, okay? They all stay in place and they're recyclable. So looking at just some basic properties, number one, enzyme always remains intact, right? They're not used up. They can continue to be used and used and used and used until maybe something happens, like, for instance, the temperature gets too hot. If the temperature gets too hot, then the enzymes don't really work. So in our lab, we had a cooked potato and a raw potato. And you should have seen that the hydrogen peroxide didn't work as well with the cooked potato, because when you increase that temperature in the potato and you cook it, all those enzymes, that catalase in the potato, they start to change shape and they're not able to break down the hydrogen peroxide. So again, what you should have seen is in the cooked potato, the reaction not happen as fast when you poured hydrogen peroxide on it. Another instance is specific pH. If it gets too acidic or too basic, enzymes generally won't work. And again, when enzymes don't work and they change shape, that's the term we call denature. And enzymes are important because they control everything in our body. So I hope this helps. I hope this helps with answering your lab questions. See Mr. Matthews with any other questions.